So when we say heredity, this is the process of transmission from of the genes of parents going to their offspring. And when we say variation, this is the difference that we observe among individuals. Since all individuals are different from each other, we are unique, consider it as a form of genetics. So, in everyday life, ang ating differences, laging involved dyan ang genetics. Kasi nga, genetics is the study of heredity and variation. Okay, always remember the two keywords, heredity and variation. For the history of genetics, how was it explained by our ancestors? From their perspective, there are several hypotheses and theories how our ancestors explain the transmission of genes from parents to offspring. But disclaimer lang po, during the primitive times, walang terms such as genes, walang terms such as DNA, chromosomes, or other terms that we are using right now. So they are most likely using terms such as factors, characters. Yun lang po ang ginagamit nila. Kasi nga, wala pa namang proper terms for that during their time. So one of the hypotheses that they presented is what we call the blending hypothesis. So when you see blending hypothesis, from the name itself, blending, the genetic material of the offspring is a blend of the two parents. So for example, most likely, our ancestors based this hypothesis sa flowers. So di ba nakita na kasi dati na pag flowers, kapag red and white, the flower na pinagsama, possible kang makakuha ng pink na flower. Although, this blending hypothesis has also inconsistencies. Kasi minsan, kapag nag-cross ka ng red and white flower, possible na makakuha ka pa rin ng red flower. Or pwede ka rin makakuha ng white flower. But in the blending hypothesis, what we are explaining is that always na ang genetic material ng offspring is the blend of the parents. And sometimes it's not applicable. Okay? So another hypothesis that they presented is what we call the particulate hypothesis. So when you say particulate hypothesis, this is where the parents pass on discrete heritable units. So for the particulate hypothesis, for them, let's say ang parent, in the human perspective, let's say ang parent ay straight hair, another parent is curly hair. So in that case, kapag yung offspring ay straight hair ang namana, so siya ay nagmana lang sa kanyang nanay. Yung genes ng kanyang tatay, walang contribution with regards to hair type. Okay? So, medyo may inconsistency din ang particulate hypothesis kasi for them, namamana mo lang yung isang trait mo galing sa isang parent. Hindi involved yung isang parent kapag hindi nag-manifest yung kanyang characteristic. Okay, for the blood theory by Charles Darwin, he proposed the traits of the parents were passed to their offspring through the blood. So for Charles Darwin, wala siyang mechanism on how these traits are being passed through the blood. He just believed that because during pregnancy, we are carried by our mothers for 9 months. So this is where the process of transmission of the genes from parents to offspring happened during the 9 months. Okay po? So that's the history of the several hypotheses and claims with regards to the transmission of the traits from parents to offspring. So, sino ba nag-disprove ng mga theory na ito ng ating ancestor? It was Gregor Mendel who provided the explanation on how the genes are passed on from parents to offspring. So, how did he do that? He carefully planned his experiment with garden peas. So Gregor Mendel was an Australian monk, and then he is staying in the monastery. So during his time in the monastery, he was able to conduct an experiment with garden peas, so that he he can explain on how this process work, yung mechanism of transmission ng traits from parents to offspring. What did Mendel do again with this experiment? He really planned for this experiment so that he can really explain the concept behind the inheritance. So for Mendel's approach, his methods allowed him to explain the principles of inheritance, but somehow it remains elusive to others. So when we say elusive, some people really don't understand the principles that he presented. Why? One of the reason for that is some of the people, they are still sticking to the traditional explanation of the primitive people. So some of them are still believing yung blending hypothesis. Some of them are believing the particulate hypothesis, even the blood theory. So kumbaga, close-minded sila na may bago explanation, merong mas plausible na explanation. That's why others cannot understand the principles of Mendel. Isang reason din naman why others cannot understand because the lack of background. And most of the people around Mendel, they were able to understand ano ba yung mga pinagsasabi ni Mendel. And then during his experiment, he was able to observe a heritable feature that varies among individuals, which is what he referred to as character. So ngayon, sabi ko nga kanina, wala pang terms na mga genes, wala pang terms na alleles during his time. So yung term ni Mendel for the gene ay character. Okay? So when we say genes, ito yung mga heritable feature that varies among individual. Ang kay Mendel na term ay character, ang term ngayon ay genes. Okay? So, kumbaga, baka malito kayo, ano ba yung gene, ano yung trait? When we say trait, the variant of each gene or the variant of each character is called trait. So, example, kumbaga, ang category, siya ay gene. Example, gene for flower color. So, siya yung gene. Siya yung category. Ano yung traits na involved dun sa gene? Yun yung variants. Ano ba yung variants ng flower color? In terms of Mendels experiment, merong purple at merong white colored flower. Okay? Another example, kung sa human perspective naman, kung ang category ay hair type, yun yung gene. Ano yung variants ng gene for hair type? Ano yung traits? Pwedeng straight hair, pwedeng curly hair, pwedeng wavy hair. Okay? So, para hindi lang kayo malito, ano ba yung gene at saka ano yung trait. And if you have read our materials in bio, you will I hope that you have read na yung piece meron siyang 7 characteristics that Mendel observed. And these characteristics helped him to become successful with his experiment. And then, how did his experiment become successful? With regards to the use of garden peas. Kasi isang factor why his experiment was a success is because of his choice of test organism. Okay? So when you say test organism, this is the organism that you are studying in your experiment or in your research. So Mendel really planned for, really choose the garden peas kasi sobrang convenient niya. Another advantage of using garden peas for his experiment, it has a short generation time. So when you say short generation time, you can, di ba sa research, kailangan meron kayong mga replicates. So, bakit kailangan ng replicates? Kasi, para mapatunayan na accurate yung inyong data. In regards to garden peas, the short generation time of garden peas contributed to the success of his experiment kasi he can have a lot of replicates with his study. That's why his data was really accurate. Kasi marami siyang replicate dahil ang garden peas madaling mag-reproduce. Kasi yung ibang plants, when they reproduce, they take about 2 to 3 months before they can reproduce a lot of offspring. However, yung garden peas can only take 2 to 3 weeks to reproduce. And then, another advantage of using the garden peas would be it produces a large number of offspring. So, kumbaga, hindi talaga mahirapan si Mendel mag-replicate kasi yung isang generation time ng garden peas, sobrang daming offspring ang pwede niyang makuha. Madaming replicates din, maraming population. And then, for another advantage, the last advantage of using garden peas for his experiment is that mating of garden peas can be controlled by the researcher. So, Mendel can control kung kailangan bang mag-self-pollinate, kung kailangan bang mag-cross-pollinate. Isang misconception lang that I want to clear with self-pollination is that, di ba ang kalimitang turo, I don't know with you guys if you have the same background with the other students. Pag sinabing self-pollination, ang kalimitang tinuturo, isang individual lang and it will self-pollinate. Kasi nga naman, self-pollinate kailangan within yourself. So in terms of flowers, yung isang flower, kung siya ay self-pollination, dapat sa kanya lang. It's wrong. Okay? So if you have a purple flower, kailangan, kapag siya ay nag-mate with another purple flower, it is still self-pollination. You are mating with the same variant. Okay? Kung purple to purple yan, self-pollination pa rin yan. Hindi yan cross-pollination kasi ang cross-pollination is with another variant. So if you will cross a purple flower with a white flower, that's cross-pollination. Pero pagka purple to purple, even if they are two individuals, it is still self-pollination. Okay po. I would just like to clear yung misconception about self-pollination. Hindi siya isang individual lang. Possible siya na several individuals but of the same variant. And then, during his experiment, what did Mendel do? Di ba ang parts ng flower, meron tayong male and female part of the flower. So, ito ay yung stamen and pistil. So, dun sa experiment ni Mendel, he cut the portion ng purple flower, he cut it here, and then he inserted it sa carpel ng flower. Ang carpel is the female part of the flower. Okay, and then after that, he just wait for the peas to grow, and then after magkaroon ng garden peas, mag-grow ng garden peas, in-observe na lang niya yung characteristics. And then, as I have said earlier, during his experiment, Mendel only tracked those characters that occurred in two distinct alternative forms. So, ano ba yung alternative forms? Di ba kanina, sabi ko sa inyo, yung traits na trinak ni Mendel, if you will see this table, ito yung dalawahan lang. Example, seed color. Ano yung traits niya? Yellow or green lang. Kasi possible na if there will be 3 traits, may hirapan siyang mag-detect ng inheritance. Kaya nanggaling yung pangatlo or bakit nawala yung pangalawa. That's the reason why sa dinami-dami ng genes or characters ng garden peas, bakit 7 lang? Because these 7 characteristics, they only have 2 contrasting traits. And aside from that, Mendel used the true breeding plants. When we say true breeding plants, these plants produce offspring of the same variety when they self-pollinate. So for example, kung purple flower ang ginamit ni Mendel, kung true breeding plant siya, kahit mag-reproduce siya, lahat ng offspring ay purple. Wala magiging white, wala magiging pink, wala magiging red. And then yung isa naman, kung white siya, if it reproduces for several times, white pa rin ang lalabas, walang iba. That's what we call the true breeding flowers. And then, in Mendels experiment, these two, yung purple and white flowers, is what we call the P generation. When you say P generation, it is short for parental generation. Okay? So yung kanyang parental generation is composed of true breeding plants. And then, dahil magkaiba sila, a true breeding purple flower and a true breeding white flower, when Mendel combined them, there is the process which we call hybridization. And then since siya hybridization, ang expected offspring is what we call hybrids. So after hybridization, we have the F1 generation, which is short for first filial generation. And on Mendels experiment, he was able to observe only purple flowers during F1 generation. So he was curious like, bakit kaya purple flower lang? Eh meron namang magulang na white, meron namang parent na white flower. So, di ba may claim yung particulate hypothesis na nagdi-disappear yung specific gene kapag hindi siya na-express ng offspring. But still, Mendel was curious where the white, the trait for the white flower went during the hybridization process. So what he did is to self-pollinate this F1 generation. So during after self-pollination, naging successful yung self-pollination, and then it resulted to the F2 generation, which is short for second filial generation. Okay po? So dun sa second filial generation, nag-appear na uli yung white-colored flower. So Mendel was able to obtain the 3:1 ratio. Just don't mind the ratio yet. Mamaya malalaman nyo kung para saan ba yung ratio na yan. So he was just curious, bakit kaya nag-appear uli yung white? Eh di ba kayo na purple flower lang? So yung experiment kasi ni Mendel ay naka-based lang sa computations. Wala pa yung mga Punnett Square na meron kayong background. And then, wala pa rin talaga yung mga other methods on how this, during Mendel's time, how this Punnett Square works. Okay, so kumbaga si Mendel naka-based lang siya sa probability, sa computations niya, sa mga percentage ng na nakita niya noong nag-observe siya ng garden peas. Okay? And then, as I have said earlier, during Mendels experiment, gumamit siya ng true breeding white and purple flower for the P generation. And then, during the F1 generation, all the hybrids were purple. And then, since he was still curious, he let the F1 hybrids pollinate to form the F2 generation. So, yung F2 generation, that is where he saw na nag-occur uli yung white flower. So, in his experiment, one of the reasons that Mendel provided is during the F1 generation, only the purple flower factor was affecting the flower color. But since in the F2 generation, he was able to see that the white flower color occurred again. So he said that he claimed that the purple flower color is a dominant trait while the white flower color is recessive. So during his time, parang na-disprove na niya yung particulate hypothesis that whenever a gene is not manifested by the offspring, it becomes diluted or destroyed. So he was able to disprove that claim. Kasi during the F2 generation, nag-reoccur pa rin naman yung white flower color. Therefore, it was not diluted nor destroyed. And then, Mendel observed the pattern of the same inheritance in 6 other plant characters, which I have shown you earlier. And then, what Mendel called our heritable factor is what we now call as a gene. And based on Mendel's experiment, again, he was able to explain bakit ba may 3:1 ratio. Ano ba yung 3:1 ratio during the F2 offspring? So, before we discuss the ratio, let me just present to you the different concepts that Mendel was able to devise because of his experiment. So itong concepts na ito ay related to your lesson in Bio 1, which is about genes and chromosomes. For the first explanation, we have the principle of paired unit factors. So when you say principle of paired unit factors, there are alternative versions of genes which account for variations. So ano ba yung, remember kayo na yung variation ay traits. And then, this variation, meron silang alternative versions of the gene. One can be dominant, the other can be recessive. And we call these alternative versions of gene as alleles. So for example, kung sa flower color, ano ba yung 2 versions ng flower color or yung 2 traits ng flower color Yung isa doon ay purple flower color, and then the other one is the white flower color. And the alternative versions of the gene is what we call alleles. And each organism, we have 2 alleles per gene. Okay po, kaya siya principle of paired unit factors. Because each organism, we have a pair of alleles per gene. So example, kung ang gene for hair color, meron tayong 2 alleles, gene for skin color, 2 alleles din siya. Sa lahat ng genes, meron tayong tigda-dalawang alleles. Kaya siya principle of paired unit factors. Then each gene resides at a specific locus on a specific chromosome. The other concept that Mendel presented from his experiment, so as I have said earlier, for each character or for each gene, an organism inherits 2 alleles, 1 from each parent. Okay? So lahat ng genes na meron kayo or lahat ng alleles na meron kayo, yan ay, sabi nga sa blending hypothesis, yan ay blend ng parents ninyo. Mixture yan ng genes ng parents ninyo. Okay? And amazing, what is amazing with Mendel, he was able to explain this without knowing the concept ng chromosomes, particularly meiosis. Kasi nga during this time, wala pa naman yung mga meiotic cell division during Mendel's time. Nauna pa nga ang concept ng genetics kaysa cell biology. Na inaral nyo ng Gen Bio 1. So the law of dominance, di ba kanina sa F1 generation, as a P generation, meron siyang purple and white flower. And then sa F1, ang naging result lang ng offspring ay purple colored flower. So in that case, kapag yung 2 alleles are different from each other, the dominant allele will always manifest or will always be manifested in the organism's appearance. So di ba kanina sabi ni Mendel, ang purple colored flower, ang purple color ay dominant over the white color, which is a recessive allele. So sa kanya, kapag meron kang 2 different alleles, let's say ang alleles mo ay isang dominant at saka recessive, you will always manifest what the characteristic of the dominant allele is. For the 4th concept presented by Mendel, which is the law of segregation, the 2 alleles for a heritable character separate during gamete formation and end up in different gametes. Remember yung meiosis. Diba yung meiosis, we have, this is what we call the reduction division. So, ang kanyang resulting daughter cell ay haploid daughter cells. So, yung haploid daughter cells, bakit siya naging haploid? Kasi these alleles, they separate during gamete formation. So, let me just discuss you how will you determine ano ba yung mga napo-form na gametes or ano ba yung napo-form na alleles during gamete formation. So, let's have this example. If you have this genotype, how will you determine the possible gametes that will be formed? So, if you are given this genotype, how many possible gametes? You just do it this way. I-bracket nyo muna yan. So, you have a capital A, B, and then another bracket. And then you have this one and another one. Okay? So, in this example, ito yung mga, di ba sabi ko nga sa inyo, during meiosis, reduction division siya. So, maging haploid. Instead of having 4 chromosomes or 4 alleles, you will only have 2 coming, dito sa example na to, you will only have 2. Kasi nga, siya ay nasa gametes na. So, ilang gametes ang posibleng ma-form? You have 1, 2, 3, 4. There will be 4 gametes. And then, how many possible allele combination? So, dahil sila ay unique from each other, this would be 4 allelic combination. Okay po. Another example is this one. So, if you have this genotype, ilan ba yung possible gametes na ma-form? So, try nyo lang. You have, ito yung una. Ayan. So, you have A, B, C. And then, second one, you still have the same cross. Tapos, ito naman sa maliit. So, you have A, B, and small c. The second cross, capital A pa din, tapos itong yung pangalawang B, tapos yung C. So, dalawa na siya. Tapos yung pangatlo, pang-apat pala, another repetition. And then, for the third one, kapag siya ay yung small letter na, so you have the first one, and then another cross dito. Tapos, another one, and then another one. Okay po? So, dito, ang tanong dyan, how many gametes will be formed? Kapag gametes, kung may repetition regardless of the repetition, you will count the number of daughter cells, the number of gametes that you formed from the genotype. So in this case, kahit nag-repeat po itong mga ito, counted pa rin siya as gametes. Therefore, in this genotype, there will be 8 gametes that will be formed. Okay? But how many possible allelic combination? So kapag allelic combination na, pag nagre-repeat na yung combination, you don't count the repeated combination. Hindi nyo ito i-count. That's why in this genotype, there will be 4 possible allelic combination. Due to the advancement din ng scientific thinking and due to having the scientific method, so nagkaroon ng diagram which is what we call now the Punnett Square. So in Punnett Square, always remember that the capital letter represents a dominant allele while the lowercase letter represents a recessive allele. Lagi niyong lagyan ng legend. Let's say if we are talking about genes for height, so yung capital T is for tall, and then yung small t is for short. Before we discuss how Punnett Square works, ito muna yung vocabulary para well-versed kayo with genetics. So when we say homozygote, di ba sabi ko sa inyo kanina, each organism has 2 alleles, one coming from each parent. So if the organism has 2 identical alleles, you call it homozygote, or he is homozygous for the gene controlling the character. So example, kung siya ay, di ba yung ating legend kanina, capital T is for tall, and then small t is for short. So kung ang kanyang alleles, ang kanyang 2 copy ng alleles ay parehong capital or parehong small, then this is homozygous. He is, the organism is a homozygote. Okay? While if the organism has 2 different alleles, if yung isa ay capital, yung isa naman ay small letter, then hindi siya heterozygote. Okay? So again, kapag ang organism ay 2 identical alleles, pwedeng parehong capital or parehong small, it is called homozygote or homozygous. And then for the organism with 2 different alleles, one is capital, one is small, ito po ay heterozygote. Okay? So dito naman sa yung homozygous, 'di ba sabi ko kanina, kapag pure breeding plants, when we talk about Mendels Experiment, if there are pure breeding plants or true breeding plants, whatever happens, they will produce the same variant of the organism. This happens kapag nag-self-fertilize ang homozygotes. When I say homozygous dominant, meaning, pag sinabing homozygous dominant, automatic, dalawa ang kanyang allele ay parehong capital letters or parehong dominant allele. Okay? Yung sa dito, yung sa recessive, commonly pag genetics kasi, pag sanigan sa genetics, you don't say homozygous recessive. Kasi once an organism is recessive for that gene, automatic, yung kanyang genetic makeup or yung kanyang genotype is recessive. You don't really say homozygous recessive. Okay? So, well, for your sake, since you're still beginning with genetics, you can use homozygous recessive or you can use recessive na lang. For heterozygous, you don't say heterozygous dominant kasi in the first place, wala namang heterozygous recessive. So, heterozygous individual is just heterozygous. Okay, no need to say heterozygous dominant. Okay po? And I think nawala ata yung slide na, okay. So there are also two terms na possible na ma-encounter nyo in the Punnett squares. When you say phenotype, this refers to the physical appearance of the organism, regardless whether he is homozygous dominant or if he or she is recessive or kung siya ba ay heterozygous, regardless of their genetic makeup, kung ano yung physical appearance niya will be the phenotype. And then when we say genotype, this is where we determine ano ba yung genetic makeup ng organism. In the case of a homozygous, di ba, dito sa homozygous dominant at saka sa heterozygous, both of them are tall. Okay? So, kung tall sila, yun ang phenotype nila. Both of them are tall. However, they have the same phenotype but they have different genotypes. Possible siya. Kasi di ba kapag ka naman heterozygous, kung ano yung characteristic ng dominant allele, yun ang magmamanifest. So both of them have the same phenotype but they have different genotypes. So in the case of Mendels Experiment, yung sa purple and white flowers, di ba yung kanyang ratio ay 3:1. So 3 purple flowers is to 1 white flower. So itong tatlong purple flower, they have the same phenotype. But if you will look at their genotypes, the other one is different from the other two. Okay po? So again, several organisms can have the same phenotype, but they can also have the different genotypes. And then ano ba yung use ng Punnett Square? For us to identify kung ano yung possible gametes or genotypes na present sa isang individual, we perform a test cross. And this test cross uses the Punnett Square. Anong example ng test cross? If we have the parents' genotype like this one, so both parents are heterozygous for being tall. So, ang Punnett Square, wala naman siyang proper position kung dapat ba yung noo ay nasa taas. Wala namang ganon. Depende sa inyo as long as you can write the correct answer. So, you just use the Punnett Square. Ayan. And then write the answer. So, for this one, you have a homozygous dominant. This is heterozygous, another heterozygous, and a recessive individual. And then, from this Punnett Square, you can derive the phenotypic ratio. So, yung phenotypic ratio and also the genotypic ratio. I suggest na pagbago pa lang kayo sa genetics, you derive muna yung genotypic ratio bago yung phenotypic ratio. Okay? So, again, ang genotype refers to the genetic makeup of the organism. So, based on the Punnett Square, you have here 1 homozygous, and then you have 2 heterozygous, and 1 recessive. And then for phenotypic ratio, you just add all tall organisms and all short organisms. I suggest that you put yung legend kung alin ang tall, alin ang short. Okay, so for this example, you have 3 tall is to 1 short. Okay po. And then, disclaimer lang ha, kalimitan kasi ng misconception ng mga students sa genetics. Dahil apat yung offspring sa Punnett Square, pagka nagkaroon ng example na 5 yung offspring, ine-expect nila na, hala, miss, bakit 5 yung offspring? Eh, di ba sa Punnett Square, 4 lang? Let me just emphasize that the Punnett Square will only tell you the genotypes and the chances of getting those genotypes. And hindi rin ibig sabihin na sa magkakapatid, let's say sa Punnett Square, may 4 na offspring. Aside na hindi given na 4 lang dapat yung magiging anak, hindi rin given na pag 4 kayong magkakapatid, 3 agad ay tall, yung isa ay short. Okay? I don't know with the, sa stat kasi ang tawag dito ay mutually exclusive events. So yung each offspring or each child in the family or each organism in the family will have an equal chance of obtaining these genes. Kasi hindi naman kayo sabay-sabay fertilize, ini-schedule lang yung inyong panganganak. Hindi po ganon. Okay? So for example, kung si panganay ay tall, hindi given na dalawa na lang ang pwedeng maging tall sa family at isa na lang ang pwedeng maging short. Again, these are mutually exclusive events. So, hindi given na laging kapag may tall na sa family, mabawasan ng chance na maging tall din yung susunod sa kanya. So, dito, kung ang tanong, pwede naman tanongin dyan, what is the probability of getting or having a tall offspring or a tall plant? So, pwede yung lagay nyo dyan ay 75% or 1/4. Ganyan. So when you say mono-hybrid cross, this would be a cross that we are only looking at one gene or a single character. Kaya siya mono, from name itself, mono means one. So we are only talking about one gene or one character for mono-hybrid cross. Kasi yung dihybrid cross, when we say dihybrid cross, we will be talking about two genes at the same time. Or we will be observing the transmission of two genes at the same time. That's dihybrid cross. So later, I will explain kung ano ba yung law of independent assortment. But let's discuss muna kung ano yung dihybrid cross. Okay, for dihybrid cross, example, ang parent's genotype would be, ang genes na ito ay round and for seed shape at saka seed color. So again, pag lagyan nyo muna ng legend. So let's say ang R ay round, and then ang recessive naman po ay wrinkled. And then for letter Y, ang dominant is yellow, and then ang recessive is green. Okay? So we have two ways on how to solve the dihybrid cross. Ituturo ko yung isang shortcut sa inyo para pagka kasi Punnett Square, you will observe later na medyo nakakahilo ang Punnett Square. Kasi kung kayo nasa mono-hybrid cross, apat lang ang offspring. Pagka siya ay dihybrid cross, 16 na. 16 na yung inyong ika-count. Okay? So, yung unang step ay Punnett Square before I teach you the shortcut. Sa Punnett Square po, what you will be doing first is perform yung FOIL method natin kanina or yung sa determining ng genotypes. Ay, ng gametes pala, sorry. So, you have to determine the possible gametes that will be formed from the genotypes. So, from the first parent, which is this one, ito yung kanyang genetic makeup or genotype. So, mag-FOIL method lang kayo. Ano yung possible genotypes? Ay, gametes pala, sorry. And then, this one, another one, another one. Okay po? And since both parents have the same genotypes, you will just expect the same set of gametes that will be produced. So, para maging Punnett Square siya, sulat niyo lang siya ng ganyan. Pababa naman yung isa. Okay po? And then, perform the Punnett Square. So, lipat lang ako ng ibang slide para may bakante. So, ano yung possible gametes? You have this one. Okay. And then, mag-Punnett Square lang po kayo. Kung ano yung given na unang letter, yun na yung isulat ninyo ha. Pagsamahin nyo yung magkasamang letter. And always remember, per gene, you have to write the capital letter first. Per gene. And then, after you have performed yung sa Punnett Square, yung test cross, syempre, you have to count yung genotypic ratio. Para hindi kayo malito, edi mark nyo siguro ng small x dito sa gilid, kung alin na yung na-count ninyo. So, you have one, Okay, so dapat po ang total ay 16. And then, phenotypic ratio, pag samasamahin lahat ng magkakatulad, yung una is round and yellow. Alin ba dyan ang round and yellow seeds? So you have 1 plus 2, you have 3 plus 2, 5 plus 4, 9. And then, next, ano ba ito? This is round and green. So yung round and green, ilan ba? You have 1 plus 2, you have 3. And then, the next one is wrinkled and yellow. So, alin dyan ang wrinkled at yellow? Ito, 1 plus 2, you have 3. And then, the last remaining phenotype is wrinkled and green. So, 9, 3, 3, 1. This is what you call the branching method. Okay, so sa branching method, so recall lang natin kayo na yung genotypes ng parent. You have this genotype. Ayan, okay? And then, kanina, kailangan nyo determine yung gametes ng parent or yung possible na gametes from the parent. So, ngayon naman, ang kailangan nyo muna, i-Punnett Square nyo muna yung unang genotype, ay yung unang set of genes. That would be the genes for the seed, alleles for the seed shape. So, yung sa unang parent, siya ay heterozygous. Sa second parent naman, heterozygous din siya. So, you just perform the Punnett Square. You have homozygous dominant, heterozygous, and then recessive. After that, you get the genotypic ratio. Ano yung genotypic ratio from the cross? You have 1 homozygous is to 2 heterozygous is to 1 recessive. Okay? After you cross yung first, after the test cross ng first gene, so you test cross naman yung second gene, which is the gene for seed color. So yung parent 1 is heterozygous as well as the parent 2. Actually, makikita kayong pattern dyan, but I will let you discover it. Kasi pag sanay na kayo mag-practice ng genetics, pag nakita nyo yung genotype na yun, automatic, alam nyo na ang genotypic ratio. Pag sanay na. Okay? Kahit di na kayo mag-Punnett Square, kaya nyong i-determine. So, ano ba ang genotypic ratio ng second cross? You have 1 homozygous, and then 2 heterozygous, and 1 recessive. Okay po? So, tandaan lang yung genotypic ratio na yan. And then, next, ay, wala nang space. Next one, you write the genotypic ratio ng first cross. So, ito ay dito sa baba ha. Okay? So, you write the genotypic ratio ng first gene, which is 1 homozygous pababa, and then 2 heterozygous, and 1 recessive. Bakit siya tinawag na branching method? Yung genotypic ratio ng second cross, iba-branch nyo siya dun sa genotypic ratio ng first cross. So you have 1 homozygous, 2 heterozygous, and then 1 recessive. Tapos, branch nyo ulit dun sa pangalawa, you have 1 homozygous, 2 heterozygous, and 1 recessive. Branch nyo ulit sa pangatlo. You have 1 homozygous, 2 heterozygous, and 1 recessive. After that, multiply nyo lang. Tapos sulat nyo yung genotype. Okay po. Actually, itong sagot ninyo dito sa multiplication na ito, yan na yung inyong genotypic ratio. You just have to copy it sa genotypic ratio. Okay po? And if you will observe the phenotypic ratio, if you count kung ilan ba yung round and yellow, so round and yellow, count lang natin, you have 1 plus 2 is 3 plus 2, 5 plus 4, 9. Tapos ilan yung round and green? So you just count it. You have 1 plus 2. You will have 3. And then for wrinkled and yellow, ilan ang wrinkled and yellow? So you have 1 plus 2, 3. Remaining, you have 1 wrinkled and green. Let me just discuss yung law of independent assortment. So kanina, we have, di ba pag dihybrid cross, we are talking about, we are observing the transmission of 2 genes or 2 characters. And that's how, even in monohybrid cross, the law of independent assortment applies. When we say law of independent assortment, it states that each pair of alleles segregates independently of any other pair of alleles. So, sabi ko nga kanina, these are mutually exclusive events. That's why your Punnett Square will only tell you the chances of having those genotypes, and also it will not tell you how many offsprings are there in reality or in actuality. Okay? So kapag ganyan, ang Punnett Square, kung 16 ang offspring, hindi ibig sabihin dapat laging total ay 16 kapag may question. Pero kapag you are doing your Punnett Square, kailangan 16 yung offspring para malaman nyo ano ba lahat ng possible genotypes. Okay? And always remember, based on the law of independent assortment, the segregation of one pair of allele will not affect the segregation of others. Hindi ibig sabihin na yung round and yellow ay laging magkasama. Pag hindi nag-segregate ng maayos ang round, hindi rin segregate ng maayos ang yellow seed, ang alleles for yellow. So hindi ganoon ang genetics based on the law of independent assortment. Thank you po. Ingat po kayo.